a very warm welcome to the third In Conversation with Ignite event. We are very, very lucky to have a conversation between two wonderful scientists today, and you'll see a few details that are on the screen, so I'm not going to give a long introduction. But we are very, very excited to have Vishva Dixit and Ramaswamy Subramanian with us. While you see a few slides kind of flashing on your screen telling you about the event, I'd just like to introduce the interlocutor for today's session, which is Ramaswamy Subramanian. He is the founder and the board member of the Ignite Life Science Foundation, who are one of the organizers for today's event. He is also a professor at the Biological Sciences and Biomedical Engineering Department at Purdue. He's a fantastic scientist, a very vocal advocate and facilitator for the life science community in India. You might have kind of crossed paths with him at other events. He's very active at engaging with the community. And so just with that short introduction of the interlocutor for today's session, I'm going to hand it over to you, Rams, to introduce Vishwa and to get started with today's session. So over to you. Thank you, Rams. Thank you, Vishwa.
Thanks, Shantala. Thanks, IndiaBioscience and others for helping us organize this. Good morning, Vishwa. I know it's 6 a.m. for you, and thank you for doing this with us. So normally I could go on a long monologue introducing Vishwa. I'm going to try and avoid that and just make even that part of the conversation. I have to say that Vishwa is one of my heroes. So about 25 years in my academic career, I have followed his work, and he's a person I would love to emulate. You know, he's such an amazing scientist and has done amazingly well. So instead of me trying to tell you about his history, I'm going to start by asking him questions about himself, and it's much better to hear from him a personal experience of his journey from being a kid in Kenya to his current position of being the vice president of Genentech, and in science going from development to death. So he has addressed issues from child development to death, and also transition from academia to industry. So Vishwa, the first question for you would be, can you tell us your journey? How did you end up here? And what were the turning points in your life that led you to be what you are today?
Well, thanks, Rams, and thanks everybody for tuning in. Well, it's been a long journey, I have to say, and we could spend the entire hour on it. But briefly, I'm of Indian origin. My parents were both born in India, and they came over to East Africa, to Kenya, in the 1940s and settled in East Africa. And my mother's from, or was from UP, and my father from Punjab. You know, I grew up at a time in Kenya as a child when it was still a colony of the British Empire. And so it was when I think back, it was sort of a surreal world, highly segregated. There you had the Black Africans, you had the Indians, and you had the Europeans, and there was a very strict segregation in terms of schools and hospitals, etc. But after independence in 1963, that went away. I think Kenya has done fairly well in terms of its progress. There were certainly hiccups along the way. But the Indian community there is probably dwindled considerably in size over the years for a variety of reasons. Many have gone to the UK, Canada, some to the US. My parents were both physicians. They were both doctors. They worked in a small town in Kenya in private practice. they were exceedingly busy, and that was something that I still remember. However, they had a lot of books around in the house, and I was particularly influenced by a series from Time Life, which described various topics, the planets, the time, biology, etc. But one of the issues was on scientists, and that stuck with me because when I read about it, it seemed that the work that scientists did, I was maybe 10 or 11 at that time, but it seemed to me that the work that they did was akin to being an explorer. And I thought that was really exciting because while there weren't too many more pyramids or countries or rivers to be discovered, here was a whole universe of discovery right in front of oneself, and one could be an explorer, and you could discover things, and your discoveries would be remembered. So it was a way to sort of immortality, and I became fascinated by it. I decided that's what I wanted to do. It's a funny thing, you decide when you're 10 or 11, that's what you want to do, and you end up doing it. That's you devote your life to it. So I went to medical school in Kenya. I qualified as a doctor, practiced medicine there, and then came to Washington University in St. Louis to do a residency in the Department of Pathology. And I have to say, in terms of influence, probably the two that stand out was my professor of physiology at University of Nairobi, Dr. Hetarachi, the Sri Lankan professor. And he really emphasized the importance of understanding science through sort of basic principles. Like he would say, you could understand physiology through Claude Bernard's constancy of the internal environment, how all organisms and systems attempt to maintain the status quo. And then you could understand how systems evolve through Darwinian biology. And those two are really the foundations of biology. Claude Bernard's constancy of the internal environment and Darwin's theory of evolution. And if you view complex problems through that lens, there'll be a simplification. And that struck with me. He would try to make complicated things simple. But when I came to WashU in St. Louis, for me, that was sort of an electrifying experience because I realized that knowledge could be challenged. We would have part of the residency program was to do research, to work in a research lab, and I worked in the Department of Biochemistry, worked on an extracellular matrix protein thrombospondin. But regardless, in this environment, and it's a very research-oriented medical school, or WashU, there was a great emphasis on scholarship, on the expectation that their trainees would end up as in the academic track. But part of that was a very rigorous research in progress series. We used to call it the Shark Tank because you had to go and defend your findings, and people would, really take a shot at you, that rip into you. But it sort of, it emphasized to me that at least at the cutting edge, how plastic the knowledge is, and we are really treading very carefully, often treading on thin ice, and it's very difficult to draw firm conclusions. And the other part that struck me was the sort of argumentative nature of research, the back and forth, the sort of pugilistic nature of the conversations. And this was sort of very different from what I'd been exposed to earlier, which was more, you read your textbook and you did exams sort of work. that really influenced me. I realized then that what's really important to succeed is curiosity. If you're curious, and I felt I was curious, then that would go a long way towards succeeding. And then questioning, the ability to question your own ideas, to pressure test them. The key is not to do experiments that support your idea, but rather to do experiments that challenge your idea. So that was, I think, a real turning point for me. I was my first introduction to real research, first introduction to thinking like a scientist, thinking critically. And then following that, I took up an assistant professorship in the Department of Pathology in 1986 at University of Michigan, and set up my own group. We worked on the biology of extracellular matrix proteins, in particular thrombospondin. And the work was going quite well, I have to say. I was well funded in the American system with R01 grants. And but I wasn't satisfied, because the question we were addressing wasn't that critical. It seemed like I was dotting the I's and crossing the T's, adding details. And I really wanted to do something larger. I wanted to have a larger impact. And so I started thinking, I started reading a lot. And it seemed to me that one has to ask a sort of a bold question. And in many cases, the question that you ask is probably the most important element of the impact you will have. And this, I would say, is important because often people plunge into a research lab and start doing work. But I think it's important to ask that if the work was to succeed, what impact would it have? Would it influence others? Would it be remembered? Would it be cited? So given that, I struggled with what to work on because I had the safety net and the surety of the grant funding on extracellular matrix. And so, but I started reading, and it seemed like the world was sort of possessed at that time in the late 80s, early 90s by the cell cycle and enormous breakthroughs happening. And I decided to think about the opposite, cell death. And in that sense, I was thinking about it at a fortunate time, because the work of Peter Kramer, Shigi Nagata, Dave Goodell, they had described death receptors, receptors like FAS and TNF receptor, which under certain situations, when stimulated, would induce cell death. And I thought, well, now here's a system. This is experimentally amenable. You could take cells in culture, you could stimulate them, and they would die. And then you could say, hey, what's the mechanism? What's the machinery of death? And that's when I embarked on working on cell death. I had some very talented students, postdocs, Arul Chennaiyan, Munish Tiwari, the MD, PhD students, who really were working very hard, were very creative young scientists, and helped launch my career in cell death research. And then in 97, I became a tenured full professor. Things were going quite well. We really did some of the best work in my career was during those days. And in 97, I moved to Genentech. And I've been at Genentech 25 years. So I'll stop there, and that sort of is a brief summary of my career.
Well, it's wonderful. So I'm going to ask you a whole bunch of questions, and these are most of the questions I'm asking are questions that are also from the audience. Almost all the questions I'm going to ask you. So you worked in academia, you worked in industry. So one of the first questions that comes to my mind is, what are the cultural differences in doing research in academia versus in industry? And is there anything special about Genentech that's different from a regular pharma company that makes it, that made you stay there for 25 years? Because you could principal have gone anywhere and done anything. So tell us about the cultural differences and how you thrived in two different cultures.
No, that's a very important question. And I'll say a couple of things. People often ask me, what's it like to work in industry, or what's it like to work in the pharmaceutical biotech industry? And I have to be honest, and I say, I really don't know, because I work at Genentech, because it is such a special place. And so I think when people ask, what is it like to work in industry, it's important to realize that industry, the biotech industry, the pharma industry, is a very heterogeneous mix of cultures, and there are many ways to skin a cat. Unlike in academia, where successful departments aspire to excel in teaching and research, in the pharmaceutical industry, really you have to make or generate drugs that will make profit, and there are many ways to do that. So you could say, we're not going to do research, we're going to acquire companies. And there are big pharma that's their model, and they're very successful. Or you could be at a very small biotech. You could be at a biotech of just half a dozen people. The culture there would be very, very different from big pharma. Or you could be a place like Genentech, which is sort of a hybrid between academia and industry, where we have a strong emphasis on research. We devote 10% of our budget, the Genentech budget, to pie in the sky, wild blue yonder research. And that's mostly done by postdocs. So we have an active postdoctoral program at Genentech. We have 140 postdocs. And condition of being a postdoc at Genentech is you're not allowed to work on a product-related project. You have to commit yourself to basic research, because the way the program is assessed is through its publication record. And that's the Genentech culture. It's a culture I'm attracted to. The sense is that science, research, drug discovery is not an engineering project. Really, there is enormous space for serendipity, for exploration without a blueprint. And so if you look at big advances, you look at restriction enzymes, or you look at CRISPR, these are revolutionary tools, but they really are part of the innate immune system of bacteria against phage, and they came out from very basic fundamental research. So there's that appreciation for that. So the culture is quite unique, I would say, and quite different, with a heavy emphasis on research. We spend just to give you an example of the figures I'm talking about, Genentech is a wholly owned subsidiary of Roche. Now, Roche is a very big company. It has a massive R&D budget. Approximately $10 billion a year is spent on R&D. And a lot of that is, of course, spent on development, because clinical trials are very expensive. But still, that's a massive investment in R&D that Roche makes. And Genentech is a subsidiary of Roche, benefits from that. And we devote, I would say, approximately 10% of our Genentech budget to basic research through the postdoc program. So that, and a number of homegrown discoveries, Herceptin with the anti-HER2 for breast cancer, anti-VEGF for macular degeneration and cancer. These were all discovered at Genentech. So it's certainly a culture and an approach that has paid off, but that's just our culture. And that's not to say that other companies follow the same game plan or rule book, playbook. They have their own ways to succeed. So there are many ways to skin a cat here. So for me, coming from academia to Genentech wasn't that big a transition because of this emphasis on basic discovery, emphasis on doing really important science. I think what I realized is that the enormous effort it takes to make a successful drug, at the end of the day, it's nothing short of a miracle. And it's the work of many, many people. And you really, to succeed in industry, you have to be a team player. You have to work well with others. You have to be able to communicate. Because there are so many disciplines that are brought to bear, from basic biology, chemistry, regulatory, metabolism, clinical trials, statistics, that eventually lead to the launch of a drug. And that's, it takes many years, it's very expensive. I would say those are the differences for the members of the audience thinking of industry. I just want to emphasize again, you can roughly think of it as sort of big pharma, where your job is, I would say, relatively stable, much more stable. You are part of a much bigger machine. You know, you have to work well with others. It's and the other extreme, of course, is startup biotechs, where there's a lot of excitement, but a lot of pressure. You're running on a short runway. You have to make it succeed. There's great need to be focused. And, you know, you're really, if you hit it big. And if you don't, you don't, right? So that's a very different culture. In a startup, you know, you're going to work really obscene hours and long days. Whereas in big pharma, it's more episodic in terms of your effort. There'll be times when you'll have to work really hard, but I would say the runway is much longer. It's a much more stable position. And where you go is really your temperament. If you're young, you're entrepreneurial, you want to, you think you have what it takes, you know, maybe a startup is the right place. If you feel you work much better as part of a larger team, you want more stability, then maybe pharma is something you should look at. So I think it's an individual choice. You know, as they say, one man's meat is another man's poison. So you've got to look into yourself and say, you know, what am I really suited for? So that's my thoughts.
Yeah, so that's fantastic. And I have a few follow-up questions, but I'm going to switch between questions about science that were asked and questions like that. So one of the interesting science questions that has come up is that, okay, you know, you worked on innate immune response, cell death, but when people think about immunology, they think normally about antibodies, but that's not where you are. So can you tell us a little bit about your research on cell death? And a provocative question is that if we can stop programmed cell death, is mortality deniable?
Yeah, well, I think when you think of, there are two aspects to that. There's the homeostatic function of cell death, and then the role of cell death in inflammation and immunology. I just comment first on the homeostatic role of cell death. You know, I often say we live because we constantly die. And a really surprising statistic is that every second, 1 million cells die in our bodies. That amounts to about just over a kilogram every day. So you lose about 2.5 pounds of cells every day. That is an enormous, enormous number. Now, of course, those are replenished with new cells. And the reason you lose a kilogram of cells every day is that a lot of them are old. Often they carry dangerous mutations. They have lost their ability to carry out their normal function. And this is the system rejuvenating itself, right? So this is a rejuvenation. And this cell death, referred to as apoptosis, is a silent cell death. You don't notice you lost a kilogram of cells every day because A, they're replenished, and B, the consequence of that was immaterial, right? It was silent. It was immunologically silent because those apoptotic cells were engulfed by the reticuloendothelial system, by neighboring cells, and they just disappeared into the night. So now that concept of cell death and replenishment is critical for all multicellular organisms. Right? But the other form of cell death, of course, has to do with the innate immune system. And remember, a major threat to us is intracellular pathogens, toxins, and the innate immune system are the first responders to that, right? So it's a very primitive system. It's present in, as I just mentioned, it's present in bacteria. That's how they defend themselves against phage through restriction enzymes and CRISPR and mechanisms such as that. So that's a very ancient system. In that particular instance, the replicative niche for the phage is denied because the bacterial cell dies. When you have situations where the innate immune system gets activated in bacteria, and I'll come back to that later. In the plant kingdom, the only defense is the innate immune system. There is no adaptive immune system. There are no antibodies or cytotoxic cells. And so I think you can think of the innate immune system briefly as the first response you have to injury. And that injury can be a burn, it can be a cut, it can be an infection. And that first response mounts and orchestrates an inflammatory response. The influx of neutrophils, mononuclear cells, cytokine production, fibroblast activation, the whole orchestration of getting rid or killing the inciting pathogen, and then instituting a healing repair process. So that's the innate immune system. And of course, the adaptive system, which is very important, takes its cues from the innate system. So if the innate system doesn't alert the adaptive system, then there is no adaptive system. There is no antibody generation. So I'm putting it very simply, but very simply, the innate system is the foundational system for immunity and healing in multicellular organisms. And in higher organisms, in vertebrates, where we have an adaptive immune system that can give you antibodies and cytotoxic T cells, it is still dependent on the alarm that is released by the innate immune system. And the alarm is cytokines and chemokines and other factors that will bring the adaptive immune system to bear. So that's the function of inflammation innate immune system. Now, a very important part of innate immune system is cell death. And we refer to this often as necrotic cell death, which in contrast to the apoptotic death that's immunologically silent, necrotic death instigates, propagates an inflammatory response because dying cells release mediators of inflammation. Those mediators can be entities like ATP, or they can be cytokines like IL-1 alpha. And part of that for mononuclear cells is activation of an apparatus known as the inflammasome, which leads to the generation of IL-1 beta and IL-18. The details are less important, but to say that you get cytokine generation and you get cell death, and both of them contribute to the inflammatory response. Now, very recently, we discovered that that many forms of death and necrosis are mediated by a pore-forming protein, Gesdermin D. And now I want to hark back to E. coli, because there was a paper in Science this year that showed that bacteria have a Gesdermin-like molecule, and that is in an operon that's activated on phage infection. So when bacteria are exposed to phage, there are many defenses, as I mentioned, restriction enzymes, CRISPR, but here we see death as a defense, where the operon is activated, the bacterial Gesdermin is engaged, it makes a pore, and the bacterial cell dies, denying the virus of a replicative niche. So this principle that I just enunciated, where the host cell dies, a necrotic death, and it denies the intracellular pathogen, viruses, intracellular bacteria of a replicative niche, is very important, and is, I would say, one of the pillars of immune evolution, is to deny the replicative niche to intracellular pathogens. And the way to do that is to have the host cell die. Now, obviously, with these mechanisms, if you were to inhibit cell death, the question of immortality, superficially, it may seem that that would be an attractive proposition. But when you think of it, you realize that if you inhibited the death of mutant cells, for example, you'd be at a great risk of cancer emerging. So I think really this turnover, rejuvenation is really essential. And inhibiting cell death, there are probably therapeutic areas where you want to inhibit cell death, but probably those settings are relatively acute. But chronic inhibition of cell death, I think the risk of that would be cancer.
So we talked about Herceptin, we talked about cancer. I was going to talk about something else, but I'm going to continue on the science, because this is interesting. Because immunotherapy for cancer is the growing field. You talked about Herceptin, and you and Genentech are kind of leaders in this business. So the question is, how close are we to immunotherapy becoming the mainstay in cancer treatment, and that cancer becomes something that we can routinely treat? Even today, cancer is the biggest killer, one of the biggest killers. So when will we have a thing that it's like what antibiotics did to infection, immunotherapy will do to cancer?
I think it's still an aspirational goal. I would say that immunotherapy has been an amazing advance, not only because of the benefit it has bestowed on patients, but also it has underscored that the immune system does have the capability to fight cancers. That's something that was debated a few years ago. So I think that's very important. And that says that therapies based on engaging the immune system should be pursued and may well be quite promising. Now, that being said, of course, most cancers don't respond to immunotherapy. And that's, I think both the disappointment and the challenge, is that can we get more cancers responsive to immunotherapy? We refer to them as cold cancers because they're very poor in immune infiltrates. Can they be activated? And I would say that's a huge, huge area of investigation in academia and industry, is how do we broaden the impact of immunotherapy. But in terms of cures, I would say that, I never thought I'd see it in my lifetime, but if you have metastatic melanoma, I mean, that was a complete death sentence. And but today, for those who respond, you can use the word cure. And there was a very recent report on rectal cancers that have mismatch repair, which are also well known to be very immunoresponsive, and just an amazing outcome. I think we are talking about cures in these cases. So I think the excitement is twofold. One is that when it does work, which is in fairness, in only a few cancers, melanoma, mismatch repair, certain other cancers, it's profound. You can talk about cures. But I think the challenge is that the vast majority of cancers are still unresponsive, especially to the same extent as melanoma, for example. So I think that's the challenge. But, I would say that there are advances being made when you think of cancer. You can't forget the other areas also that are the mainstay, surgery, radiation, chemo, targeted therapies. And I think for a country like India, a very important part of, when you think from a societal viewpoint, is actually prevention, whether it's environmental, from betel nut chewing to particulates, whether it's infection, hepatitis, Helicobacter pylori, stomach cancer, so you've got HPV, and now you have a vaccine. So you've got a lot of preventative cancers, where changes in lifestyle can really lower one's risk. And for all countries, that's an important aspect and is often looked over.
There are so many questions about careers, prospects, industry, academia. So as much as if it was just me interviewing you, I would only do science, but I have to respect the audience questions. So I'm going to switch you over to a lot of questions that people want to know answers from you. One of them is there's a constant peer pressure because most people get their PhDs in academic institutes, that somehow going to industry is a failure to be an academic. And you and I know that's not true, but how do you convince young PhDs that an industry career is exciting and as rewarding as being an academic, or more rewarding than being an academic, however you want to phrase it?
Yeah, I would say that my earnest advice would be to avoid peer pressure. It can be a huge influence, but it often leads to herd behavior. You're doing it because your friends are doing it. And if you look at recent examples of people who are eminently successful, Bill Gates never got a college degree. Elon Musk dropped out of his first day of his PhD in computer science at Stanford. So you really have to be your own person and liberate yourself from the shackles of peer pressure. So remember that peer pressure is an imprisonment, of the mind. And you have to insulate yourself from that. And you have to ask yourself, what are your interests? What are your strengths? You got to play to your strengths in life. If you've got great strengths in mathematical thinking, you enjoy doing statistics, then maybe, bioinformatics is something for you. If you've got an entrepreneurial streak in you, you don't want to work for others, you want to start your own outfit, then it's a different career track. So I think instead of listening to peers and running with the herd, you really want to listen to yourself. You really want to have that conversation with yourself and say, what are my strengths? What are my weaknesses? What is my ambition? And based on that, you should act. And if you say based on that I really want to open up a salt mine, then you should do that because that's what you're excited about, right? Whereas with peer pressure, you're susceptible. There's really the danger of running with the herd.
So there's a very interesting question, which in the vantage position you sit in, I think you're apt to answer. There is this idea of this large data collection biology, big biology, as we call it, sequencing thousands of genomes, doing large-scale metabolic profiling. And then there's this hypothesis-driven, very focused research that one does. Now, are they in conflict with each other? How does one manage both? And is it a dichotomy or both? How do they come together?
I think it's a tool. And, I've been around long enough to see a lot of very important tools come and make an enormous impact on science, but also be prone to abuse. When I think of when monoclonal antibodies came around, when cloning came around, people were stamp collecting, helter-skelter, cloning this and cloning that just because it could be cloned, making an antibody to this, that, or the other, publishing it. There wasn't a focus, there wasn't a depth to the work. And, computational biology, the databases, bioinformatics is a fantastic tool for the questions you have, but it'll always be garbage in, garbage out. None of these tools will make a dumb question into a brilliant answer, right? So I think more importantly, instead of doing checkbox sort of research where you say, well, I can do total RNA-seq. Oh, I could do single-cell RNA-seq. I'll do single-cell RNA-seq. Hey, I could also do ATAC-seq. Okay, I'll do that. You have to sit back and say, why am I doing it? What am I going to learn at the end of the day? Am I just going to produce a diagram that's going to have nice colors, pretty wallpaper? Will it really inform me? And I think you have to take that view of your work. And that's why you have to be very sort of self-critical of what you're doing. And I think if you've got colleagues, good friends who are well-wishers, these people are not going to tell you how great your ideas are, but are really going to challenge them and pressure test them. And I think the best science is done when people are asking each other and those difficult questions like, why exactly are we doing single-cell RNA-seq? Why do you want to do spatial transcriptomics? What is it going to tell you that immunohistochemistry won't? And I think once you ask those difficult questions, then you just start doing better science, So I think that's just an important part of being successful.
Yeah, that's nice. I mean, what you say is like, yeah, it's not big science is not different, but if the questions of collecting large amount of data justifies the questions you're asking, then do it. Otherwise, it's kind of meaningless stamp collection, and it's not no use. I mean, I think there is no dichotomy. It's a tool.
They asked Maurice Wilkins, who with Rosalind Franklin had the diffraction pattern for DNA, that, listen, how come you never did the structure? And he said, well, I and Rosalind Franklin, we had a strained relationship. We really didn't discuss our science and have a counterpoint discussion. He said this was in stark contrast to Watson and Crick, who argued to the limit of their friendship. And I think it's this sort of argumentative nature, questioning nature, challenging nature that really allows good science to be done. Why are you doing this? Why are you addressing this question? Who cares if you answer it? Will it have an impact? Yeah. Will bulk RNA-seq give you the same answer as single-cell RNA-seq? I think that is very important. So I don't think computational biology is a panacea, but rather a tool that we should use, do use, a powerful tool, but it'll never replace a good question.
Fantastic. So again, I'm going to switch. You visited India a number of times. You were very familiar with Indian academia and industry. So there is another interesting question that has come up is, when will India have a Genentech?
Oh, I think India has such enormous potential, is so many young people, bright people. I think in many ways, there is a liberation now with the private sector, but a lot of Indian ingenuity and entrepreneurship and brilliance has been imprisoned by a stifling bureaucracy, which is beginning to change. And I think when you say, will there be an Indian Genentech? I think, of course, I think that, but it'll require certain changes. Some have to be at a macro level, like you have to incentivize research and development. For example, it has to be a tax benefit to companies to do that. So I think there's an entire legislative aspect to it. In the pharma arm, your regulatory branch has to be very efficient, has to be a neutral entity that's not influenced by private or government things. So I think there are things at the macro level that need to be done to incentivize companies investing in R&D. That's very important. The other thing, of course, is that you have to ask yourself, what are our strengths? And in India, vaccines is a big strength, and chemistry is a big strength. So you could say, well, okay, now we are the vaccine suppliers of the world. Through our chemistry, we are doing a great job in generics. But now let's go to the next step. For the vaccines, let's go into nucleic acid-based vaccines, the future, right, of vaccinology. In generics, let's try to take our talent and ability in chemistry and maybe go into an area like antibacterials, which is a looming challenge, but we in the West aren't investing enough in that area. And that would be a huge benefit to India, to the rest of the world, if we had a new generation of antibacterials. So I think if you start thinking like that, what are my strengths? What are my constraints? What does the Indian ecosystem provide me? And then capitalize on that, then yeah, you're going to do very well. I don't think you want to copy a Genentech onto India, of course, because it's apples and oranges, but there's no reason why companies like that in the pharma industry. And again, kudos to India, has done very well in generics, very well in vaccines, excellent job. But I think it's time to step up to that next level of doing innovation. So you need, again, as I said, you need the government to step in and help, not give you more regulations, but give you financial incentives to invest in R&D. And then, people like in this audience, young people have to say, I'm going to give it a shot. Entrepreneurship. And of course, that again gets to the VC community. And again, they've really been the fuel for innovation in states like California. They have provided the gasoline for innovation, but they have financial incentives to be able to take those risks. So you need a venture community that's going to be, have the ability to take some risky investments. And all of that has to be incentivized, right? Because if you don't incentivize it, people are not crazy. They're not going to do it. But yeah, I have huge hopes for India to make major inroads. I think all the ingredients are there. I think a more rapid privatization of various of the sectors I mentioned. I think that's the way to go. I think government really needs to foster, facilitate, and not barricade these changes.
So the other question that has been asked by several people is that, and I know this for a fact, my entire lab is funded by industry. I don't have an NIH or an NSF grant. I work only with industry. But it's not common to have industry-academia relationships. What are the challenges in having industry-academia relationships where they work together? Preferably, I know you're very familiar with the US context, but I don't know how much you're familiar with the Indian context, but if you can relate that to the Indian context as to what are the challenges that could be and how does one overcome that? And if you're a young academic and you want to work with industry, what are the things that you should think about?
Yeah, again, I think it's relates to my last answer, is that you really have to look at things at a macro level and incentivize that, right? So here in the Bay Area with our institutions, Stanford, UCF, SF, and Berkeley, we have an understanding where we share reagents. It's very easy to set up collaborations. We have a standard template that we've all agreed on, right? So there's no weeks of negotiation. It's been agreed to. You just sign on the dotted line. So I think you have to first realize that if it's very easy to impede these relationships, to put a brick wall between industry and academia. And it's very difficult to break down those walls, but it's worth the effort. Because once you do, you really tap into the wisdom of an enormous community. To the extent now that we have a joint postdoc program with Stanford in chemical biology. So they get to work both at Genentech and at Stanford. And that's, when you think of it, that's very forward-looking. There are obviously some risks to that, but we've said, forget the risks. The advantages far outstrip that. And the relationship, what we have in California, exists on the East Coast in America, this sort of free exchange between industry and academia, people, ideas. Europe is just catching up to that now. I mean, they're doing a good job, but they too had really very strict boundaries between industry and academia. And I think at the end of the day, if you ask yourselves, what are we pursuing? And we're pursuing knowledge for the benefit of mankind, then it really makes no sense for us not to work hand in glove. But those administrative, legal challenges, they have to be knocked down with a sledgehammer. And it takes a lot of work. So there has to be, at the level of the Indian universities, research councils, they have to say, okay, we're going to have a template, for example. You got to simplify the process. You got to have the fewer signatures you have on the document, the better you are. You got to simplify that process, foster that exchange. And then I know DBT has tried that in the past and really incentivize that. You know, say if you as a company accept students, you know, we will pay for half of their internship, whatever it is, you have to incentivize the system. But first you have to realize that it's really, really important. And the system will be at a disadvantage to others if it continues to have boundaries between industry and academia.
We have 2 minutes, so I'm going to ask you one last science question, because I want to. One of the science questions that I think is, why do we have so many ways to die? Why do cells have developed so many ways to die? And I know we have only 2 minutes to answer this, but you're the expert, so I'm sure you can do it.
Yeah, no, I sort of alluded to that earlier. I think, you know, you have the silent way to die. And the silent way to die is the way you rejuvenate your body by getting rid of old cells and mutant cells. And then you have the opposite. You have a death that is very noisy, in that you are calling in your neighbors, calling them in for a fight because you've been invaded, you've been insulted either by a burn or a pathogen, and you have to fight it. And part of that fight is not only getting rid of the pathogen, but is a repair process. And as indicated, a very, very effective way to get rid of intracellular pathogens, which is, you know, the bane of our existence, viruses, for example, is for the cell to undergo a necrotic death. So not only does it deny the virus the replicative niche, but it also at the same time, because it's a necrotic death where it's announcing itself to the immune system, it attracts immune cells, and that leads to the clearance of virus and inciting pathogen. So I think that's why there's so many ways to die. And I think that, you know, whereas we talk about programmed cell death, apoptosis, and silent, and necrosis being this loud, angry form of death, I think there are gradations in between. So when you look at a specimen, real world specimen, you're going to see both forms of death. So I think that's why there's so many ways to die. And I still think there's a lot to be discovered. It has ramifications for a lot of medicine, degenerative diseases, cancer. If we could better regulate cell death, either necrotic cell death, that when you have too much of it, you know, I think the data now for the cytokine storm in COVID, which is responsible for the mortality, it's very clear now that the inflammasome activation that leads to this necrotic death has run amok. So there, if you could regulate it in some way, that would be beneficial, right? So, and there are examples like that.
Perfect. Thank you, Vishwa. I think we have done our 1 hour. I wish we could continue, and I will definitely email you, and we should continue the conversation. There are a lot of things that are very, very interesting. Thank you very, very much for your time. We appreciate this a lot as the organizers, and I hope we can have you back again soon to continue this conversation.
Absolutely.
Thank you very, very much.
I'd love to. Thank you, everyone.
Thank you. Bye-bye.
Bye.
Thank you very much, Vishwa and Ram. Oh, Vishwa is left. But thank you very much, Rams, for moderating this conversation, and for Vishwa for sharing the stories behind his science and also a global perspective on in research in industry and academia. For those of you who are still here, I just want to make a quick announcement that these sessions happen every 2 months. So the next event is going to be happening on August 16th. It's going to be a conversation between Uma Ramakrishnan, who studies, population dynamics with a focus on mammals and even studies tigers, and Jyotsna Dhawan, who was also our interlocutor for last session. So please do block your calendars for this date. We'd love to have you back at this next conversation event. And so with that, thank you from all of us. Thank you also to the co-organizers for this event, Swami from Ignite Life Sciences and Life Science Foundation, and Smita from Cactus Communications. As always, we're going to be sharing a feedback form. Please do fill this up. It really helps us kind of, get, fine-tune these events, get an idea what are the topics you would like to hear, because these really are informal conversations on the stories behind the scientists, behind the science, and with global perspectives on science. So with that, a big thank you. Once again, thank you, Rams. And good night from all of us, or I guess good morning for those, depending on where you're joining us from across the world. Thank you.